Hello, this is Matt Dean with A Plus College Ready, and today we're going to talk a little bit about Mendelian inheritance. Gregor Mendel is usually considered to be the father of genetics. He did a lot of groundbreaking work with inheritance in pea plants. He did most of his experiments at an Austrian monastery in the late 1800s. He's credited with discovering the basic laws of inheritance, and he was able to do this before science had any understanding of DNA, chromosomes, or even meiosis. One of the characteristics of Mendel's work that made it so successful was that he did quantitative work in biology. That means numerical. Most biologists at the time of Mendel didn't think that biological processes could be mathematically analyzed. Mendel used math, did counting, did some statistics, and his main conclusions included, number one, that each trait is controlled by a single pair of alleles. He called them factors. We know there's some exceptions to that rule, but that was Mendel's basic conclusion. Secondly, he said that some alleles are dominant and can mask the appearance of recessive alleles. That's his law of dominance. Thirdly, the alleles from a pair separate or segregate during meiosis, and only one member of each pair is transmitted to an offspring from each parent. We call that the law of segregation, again, separation. And again, remember, it's really important to remember that Mendel didn't know about meiosis, but he was essentially predicting its existence. Lastly, the maternal and paternal alleles or factors from each pair assort independently from the alleles in the other pairs. All possible combinations of alleles can occur in the gametes. We know today that this is independent assortment and that it occurs in metaphase 1 of meiosis. And Mendel called this his law of independent assortment. Some important terms to be familiar with. A trait is just a genetically determined characteristic. Again, remember that Mendel hypothesized that each trait is controlled by two alleles, one from each parent. Again, we know there's some exceptions, and we'll see that in our next presentation on non-Mendelian inheritance. An allele is simply different versions of a gene. For example, there might be a gene for eye color, but different versions of that gene might be for brown or blue or green. That's a little simplistic, but different eye colors. Dominant alleles tend to be represented with capital letters, while recessive alleles are represented with lowercase letters. Homozygous is another important term to be familiar with. An organism is said to be homozygous or purebred for a trait if the two alleles it possesses are identical. So that could be like a big T, or little t. Heterozygous is an organism that has two different alleles for the same trait. We also use the term hybrid to refer to heterozygous organisms. The phenotype is like the physical traits or physical appearance of an organism. So tall, blue eyes, type A blood. Whereas the genotype, and you don't see genotype on this slide, but genotype, those are the actual allele combinations that an organism has, like big T, or big T, or little t. So the actual gene combinations. And you see that on this slide. There's also a thing called the test cross. So let's say that in pea plants, having tall stems is dominant. So we'll use that big T to be tall. And having short stems is recessive, little t. So let's say we have a tall plant. That tall plant could be big T, or it could be big T, little t. By looking at the plant, we don't know which it is. Their phenotypes are the same, even though their genotypes could be different. Well, let's say we needed to know the genotype. We needed to know if our tall plant was homozygous or heterozygous. The easiest way to do that is to do what's called a test cross. That's when you take your plant or organism of unknown genotype and you cross it with an organism that is homozygous recessive for that trait. So if we crossed this plant, big T, with a little t plant, all the offspring would be big T, all would be tall. We shouldn't get a short organism from that cross. On the other hand, if we crossed the big T, little t tall plant with a little t, homozygous recessive, we should get half our plants being tall and half our plants being short. So that test cross will allow us to determine the genotype of an organism when we know the phenotype only. You guys have all seen Punnett squares before, and you know that those are charts that show the possible genotypic outcomes for a cross. And we'll do several Punnett squares during this particular unit. So here's just some things to remind us of the terminology. Purple flower color is dominant in this particular plant. So the phenotype is purple flowers or white flowers, whereas the genotype could be homozygous dominant, could be heterozygous. Either one of those is going to give you the same phenotype of purple, or the genotype could be little b, and that is going to give you the phenotype of white flowers. And again, this sort of summarizes it. These all have a purple phenotype, and these are the possible genotypes that will get you to that purple phenotype. This one has a white phenotype. This is the genotype that will get you to a white phenotype. We might say that this plant is homozygous and recessive, whereas this one up here is homozygous and dominant. And these in the middle, these are heterozygous. So here's our test cross. Again, in this case, we're looking at purple flowered plant. So remember, to be purple, it could be homozygous dominant, could be heterozygous. We crossed that plant with a homozygous recessive, in this case, a white flowered plant. If the plant is big P, all of the babies turn out purple. If, on the other hand, the plant is big P, half the babies turn out purple, half turn out white. All right, so now let's talk about some crosses. The most simple kind of cross is called a monohybrid cross. This is a cross in which we're looking at only one trait at a time. So to complete the cross, we must first determine the possible gamete combinations of the parent. Remember that gametes have to be haploid. They should only get one allele for each trait. So if we cross two purple flowered plants with the genotype big B, little b, and I don't know why I used B's here instead of P's, but that's okay. So big B is purple, little b is white. Both of our plants are big B, little b. By the way, the X means we're crossing them with each other, we're mating them. Remember that during meiosis, these alleles from the same pair are going to segregate. So right now, these plants are diploid for this trait. During meiosis, these two B's over here are going to separate. Half the gametes are going to get a big B, half are going to get a little b. Same thing here, half are going to get a big B, half are going to get a little b. So we have two possible gametes for each parent, either big B or little b. So we're going to draw out a square, and the square is going to be a 2 by 2 matrix, 2 columns, 2 rows. We're going to put the gametes from one of the parents across the down the left-hand side, one gamete per box or one gamete per row, like so. And we're going to put the gametes from the other parent across the top, one haploid gamete per column. And then we're just going to fill in the grid. So this first box is going to end up being big B and big B, big B, oops. So this one's going to be big B, little b, big B, little b. Always write your capital letters first. And then this last one's going to be little b, little b. So those are the genotypes. So I could do what's called a genotypic ratio. And I could say, okay, there's going to be one that is big B, homozygous dominant. There are going to be two that are big B, little b, heterozygous. And there's going to be one that's little b, little b. So that is the genotypic ratio. I also could do a phenotypic ratio. Remember, phenotypes are physical appearance, what they look like. In this case, purple is dominant. So all three of these have a dominant allele. They're all going to look the same. So my phenotypic ratio is going to be 3 purple to 1 white. And again, that is the phenotypic ratio. So here we can see a more polished version of that cross. Let's talk about probability for a minute. Remember, probability is the likelihood that something will happen. So we might say, what's the probability that this cross will produce a white flowered plant? Well, that probability is 1 out of 4 total, or we could write that as 25%. I would prefer in my class that you do these as fractions, but percentages are the same thing. So if we ask what's the probability of getting a purple flowered plant, well, that would be these 3 guys here out of 4 total, 3 out of 4, or 75%. If I ask what's the probability of getting a heterozygous purple flowered plant, well, that would be only this one and this one. So that would be 2 out of 4, or 1/2, 50%. Now, suppose that I ask, what's the probability that you get 2 purple flowered plants in a row? Well, to make that calculation, you have to use what's called the product rule. And the product rule states that in order to determine the probability of 2 independent events occurring together, you multiply the individual probabilities of each event. So in this case, the probability of getting a purple flowered plant in the first cross is 3 out of 4. Well, the first cross and the second cross are completely independent of each other. So the probability of getting a purple flower plant in the second cross is also 3 out of 4. If you want to know the probability of both of those events occurring together, you multiply their individual probabilities, 3/4 times 3/4, to get 9 out of 16, which is about 56.25%. Let's say you wanted to know what's the probability of getting 3 white flowered plants in a row. Well, the probability of getting a white flowered plant in cross 1 is 1 out of 4. The probability of getting a white flowered plant in the second cross is also 1 out of 4. They're completely separate events. Same with the third time. So to calculate the events that all 3 will be white, you multiply the individual probabilities. So you end up with a probability of 1 out of 64 that you would get 3 white flowered plants in a row. So here's the product rule with dice. So let's say that you wanted to know the probability that you would get 2 4s in a row on a dice. Well, there's 6 sides on a dice. So the probability each time of getting a 4 is 1 out of 6. The probability of getting 2 4s in a row would be 1 out of 6, first roll, times 1 out of 6, second roll. So the probability of rolling 2 4s in a row is 1 out of 36. Again, that's called the product rule. You also may hear it called the multiplication rule. So the last thing we want to talk about one of the last things we want to talk about in this presentation is dihybrid crosses. So a dihybrid cross is a cross in which the inheritance of 2 traits is analyzed at the same time. So let's say we had in pea plants that big R is for round seeds and little r is for wrinkled seeds. Big Y is for yellow seeds and little y is for green seeds. So the Punnett square that we're going to see on the next slide illustrates the cross between 2 plants that are heterozygous for both of those traits. And you can see what we mean here. So that's big R, big Y, little y for both. That's a true dihybrid cross. We're looking at 2 traits and the organisms are hybrids or heterozygous for both of those traits. To construct the cross, we first got to determine the possible gametes that each parent can create. It's important to remember that each gamete should contain only 1 allele from each gene. So in this case, each allele is going to get, each gamete rather, is going to get an R allele and a Y allele, only 1 of each. So we've got to figure out what all the possible combinations are. First of all, to figure out how many possible combinations there are, let's look at each gene first. So for the big R So again, we're going to look at each gene pair. So we've got a big R, big Y, little y. So for the R pair of genes, there's two possibilities, either big R or little r. Let's write that down, two possibilities. For the big Y, little y pair, there's also two possibilities. Each gamete could get the big Y or the little y. So to determine the total number of possibilities in the gametes, let's multiply the number of possibilities at each gene. So 2 for the R's times 2 for the Y's. There are 4 possible combinations. So let's figure out what those combinations are. Remember that each gamete has to get one of the R alleles and one of the Y alleles. So they could get a big R or a big Y. That's one. Could get a big R or a little y. They could get the little r or the big Y, or the little r and the little y. So those are the 4 possible gamete combinations for each parent. Now let's say, on the other hand, that instead of being heterozygous for both traits, let's say that the plant had been little r, big Y, little y, like so. So let's determine how many unique gamete combinations there are. Well, for the R genes, there's only one possibility, little r. For the Y's, though, there's 2 possibilities, either the big Y or the little y. So let's multiply the number of possibilities from each gene pair, 1 times 2. There should be 2 possible gametes. And those 2 possible gametes are going to be either the little r and the big Y, or the little r and the little y. 2 haploid gamete combinations. So once the gamete combinations are determined, you're going to do this exactly the way you did the monohybrid cross. You're going to write the gametes from one of the parents down the left-hand side of the square, 1 haploid gamete per row. You're going to write the gametes from the other parent across the top, 1 gamete per column. And what we're going to see is a Punnett square that looks much like this. So we've got the gametes from one parent along the left, the gametes from the other parent across the top, and then we fill in the boxes, keeping our R's with our R's and our Y's with our Y's, and writing capital letters first. And you can see over here that we get a phenotypic ratio of 9 to 3 to 3 to 1. That's an important number to keep in mind. If you're doing a true dihybrid cross, if both parents are heterozygous for both traits, you're always going to get that phenotypic ratio, assuming there's no gene linkage or anything going on. So let's do a probability question or two. Let's say, what's the probability that we end up with 2 round and wrinkled offspring in a row from this cross? Well, the probability of getting round and wrinkled is, in this case, notice there's, round and green is what I should have said. So what's the probability of getting two plants with round and green seeds in a row? So round and green. So this one is going to be round and green, round and green, round and green. So 3 out of a total of 16. So that's the probability of getting 1 round and green. So the probability of getting 2 of those is going to multiply the probability of each one. So that's going to end up at 9 over 16 times 16. So 256. So that's the probability that we would get 2 plants with round and green seeds in a row. 9 out of 256. So the odds of that are pretty small. Now there are cases where you can do trihybrid or larger crosses involving 3 or 4 or 5 traits. So let's talk about trihybrids at first. So trihybrid crosses, we're looking at 3 traits at the same time. If that was the case, the square could be up to 64, could have up to 64 squares inside it. On the AP exam, they're probably not going to ask you to draw a huge Punnett square like that, but they might ask you questions like the ones that you see below. So first of all, how many different gametes, possible gametes, can an individual with this genotype produce? Well, we're going to do this exactly the way we did it with, with our dihybrid cross. Look at each pair of genes. So for the A's, there's two possibilities, big A or little a. For the B's, two possibilities, big B or little b. For the D's, two possibilities, big D, little d. And for the E's, also two possibilities. So to determine the total number of possible gametes, we're going to multiply the possibilities at each gene pair. So 2 times 2 times 2. So there are 16 possible gametes that this particular organism could make. So let's say that instead the parent had this particular genotype. Well, for the A gene, there's only one possibility. For the B gene, there's two possibilities. For the C gene, only one possibility, little c. So let's multiply 1 times 2 times 1. There's only two possible gametes here. The gametes have to get a big A. They could get a big B. They have to get a little c. So that's one of the possibilities. So again, they have to get a big A. They could, instead of the big B, get a little b, and they get a little c. So this is one possible gamete. This is the other. Now, another kind of question you might get is one like this. What is the probability that the cross between the following genotypes will produce an offspring with the genotype big A, little a, big B, little d, big E, little e? So to answer this question, we could draw a huge Punnett square, but the easiest and the fastest way to do this is to use the product rule. First, we're going to analyze each gene pair separately, and we're going to think of this as doing essentially 4 separate Punnett squares. So first of all, we want to know what's the probability that the big A, little a from parent 1 and the big A, little a from parent 2, this should say little a right here, will yield the big A, little a in the offspring. Well, so let's do that. So let's just do a Punnett square between this and this. So big A, little a gametes from the first parent, big A, little a gametes from the second parent, so the probability that we'll get big A, little a from those two parents is 1/2, 1 out of 2. So keep that in mind. So now let's think of the B's. So what's the probability that the big B from here and the big B, little b from here will give us big B in the offspring? Again, we could draw the Punnett square, which we'll do here. But when you get some practice, you'll be able to visualize this, and you won't have to draw all these Punnett squares. So big B from the first parent, big B, little b from the second parent, so the probability of getting a big B offspring is 1 out of 2 or 2 out of 4. All right, so now let's look at the D's. So we want to know what's the probability that big D, little d here and big D, little d here will give us little d in the offspring. So again, we can draw the square or we can just visualize it. Our square, if we drew it, would look like this. So again, the probability of getting an offspring with the little d genotype is 1 out of 4 for the D's. Finally, let's look at the E's. So we want to know what's the probability that big E, little e and little e will give us in the offspring a big E, little e genotype. So again, we can draw the square, or preferably with a little practice, visualize. We end up with this square. So we can see that the probability of getting a big E, little e in the offspring is 2 out of 4, or 1/2. So if we want to know the overall probability that this parent and this parent produce this offspring, we're going to multiply the probabilities from each gene pair. So that's going to be 1/2 from the A's times 1/2 from the B's times 1/4 from the D's times 1/2 from the E's. So what that gives us is 1 out of 32. That's the overall probability, again, that a parent with this genotype and a parent with this genotype will produce an offspring with that genotype. And again, that's just using the product rule. All right, that's our. Let's do one more practice problem like that. So let me clear off a little space. So let's do one that's maybe a little smaller. So let's say that we had parents that were big F, little f, big G, and big H, little h. And the other parent, let's say, is little f, big G, big H, little h. And we want to know the probability that we would get an offspring that's little f, big G, little h. So first of all, let's think about the F's. So what are the chances that these F's and these F's give us these F's? Well, hopefully you can visualize that, but if not, again, draw the square. So that'd be big F, little f, little f. And when we fill in the square, we can see that the probability of getting little f's are 2 out of 4 or 1/2. I'm going to write that right there. So with the G's, so we've got one parent who's big G, little g. So again, we can draw our square, big G. The other parent is big G, little g. Fill in the square. So the probability that we get big G is 2 out of 4 or 1/2. Write that down. And then finally with the H's, so we've got one parent who's big H and little h, and another parent who is big H and little h. So the probability that we get an offspring that is little h is 1 out of 4. So write that down. So the probability that we get all three of these outcomes, to calculate that, we need to multiply the probabilities of each pair of genes. So 1/2 times 1/2 times 1/4. So that's going to give us 1 out of 16 as the probability that this parent and this parent will produce an offspring with that genotype. And that's the end of our presentation on Mendelian inheritance. Hope that helps.